There are 4 different categories of emerging targets in oncology. The first category is oncogenic drivers. And this category includes gene alteration that lead to the transformation from normal cell to malignant cell. The second category are the gene alterations that mediate resistance to targeted therapy. The category number 3 are targets related to DNA repair. We know that most of the cancer presents some defects in DNA repair, and it's possible now to target these defects. And the category number 4 is the target on immunotherapeutics, and this includes the group of immune checkpoints. The concept of target prioritization is the following one. So you start from sequencing data, you have different genomic alteration, and you don't know which one is relevant. So in order to facilitate the interpretation of this jungle of genomic alteration, we need to know first, among all the alteration, which are the genes that are cancer-related genes, and second, what are the characteristics of a genomic alteration that could be defined as a driver and that could be involved in cancer progression. So the question is, how do we prioritize the target? The second level of prioritization is a little bit more complex. Once you have selected your cancer-related genes, what are the characteristics of the alteration that make you think that this gene is involved in cancer progression? First, if it's an amplification, what is the level of the amplification and the number of genes? Second, is the mutation activating? Do we have a biallelic loss of function for the tumor suppressor gene? And then the question that is extremely important is how many cancer cells present the alteration in the bulk of the tumor. One of the major stakes in the development of personalized medicine is to develop tools to prioritize the target. So in terms of cancer-related genes, and there are some initiatives to develop a catalog of what is called cancer-related genes. So this is a major initiative from Broad Institute, also from other places in the world, but they are really leading this field. Then the second question is, what are the general rules that define a drivers? So for this, there are several initiatives from all over the world, from bioinformatics teams or basic science teams. And these teams are generating data to better define what is a driver. So then that are the initiatives at the research level. But then there are some initiatives that are being developed to have some list of targets or some guidance for the oncologist. In between, there are some initiatives to develop some conference to better define what are the targets that are relevant for personalized medicine. And one of the initiatives will be to develop some level of evidence for target. So until now, we have level of evidence for the drugs, but the idea will be to develop level of evidence for target. So the idea here is to provide to the oncologist some level of evidence for each target, and for each level of evidence, there will be some clinical implication. For example, you have to give the drug to the patient, or the patient should be included in the clinical trial, or don't take into account this genomic alteration. So all these initiatives are starting now. So until now, we don't have any publicly available level of evidence scale, and we don't have yet an official initiative for such classification.